The overall goal of this video is to illustrate a screening cascade that allows the discovery of novel GABAA receptor ligands. The advantage of using radioligand binding, electrophysiological recording in xenopus oocytes and in rodent brain slices in an iterative manner is that the profile of the compound can be improved. Ultimately, potent, subtype selective and efficacious compounds are identified. This demonstration will be performed by Kumiko Kambara and Jenna Tonaccini, as well as Sonia and Daniel Bertrand from HiQScreen and Marie-Clemencia from Roche.
Inject 10 to 50 nanoliters of the solution containing plasmid using a glass microinjection needle with a tip diameter ranging up to 100 micrometers, mounted on a micromanipulator equipped with a pressure ejection system or with an automated injection system. To begin this procedure, keep the oocytes at 17 degrees Celsius to prevent the expression of heat shock proteins. Store the microplate in a thermo-controlled storage area. Then, dissolve the test compounds, which were tested positive in the binding assay in OR2, at 0.1 and 1000 micromolar for electrophysiological recordings, and dispose them in a 96-well flat-bottom polypropylene plate. To perform 2-electrode voltage clamp recording, place a plate containing the oocytes on the automated system. Program the automated recording system using the icon-based interface with this scheme